Good morning. So, we are doing the chapter Human Health and Disease, Class 12th, Biology. Features of acquired immunity. Acquired immunity is that immunity that a person acquire in his lifetime, right? Like vaccination and the memory developed by taking the vaccination is called as acquired immunity. So some features are there like specificity, diversity, discrimination between self and non-self memory. What is specificity? Acquired immunity is generally the specific for a specific microorganism that will not work for any other micro. If polio vaccine is taken, then it will work only on the polio virus, not on the any other virus. Diversity. So there is a system inside our body which can recognize the vast variety of microorganism that can enter inside our body. Discrimination between self and non-self. Now, the body's immune system or immunity can recognize the self. The self means the body or the tissues of the body, and non self means the foreign tissues or the foreign particles that just entered into the body, and ability to give the response according to them. Body has the ability to give the response according to that one, right? So, discrimination between self and non self is there. कि क्या मेरी अपनी बॉडी का है और क्या कौन सी चीज़ बाहर से एंटर हुई है, कौन सा पैथोजन. नाउ memory is the feature of acquired immunity. Like whenever any pathogen enter inside the body, so body takes longer time to recognize this pathogen and to give the response. This is called primary, and if the body will give the response for that one, so this is called a primary immune response. But the memory of this encounter remain in an immune system. Whenever the further pathogen enter the second time, the same pathogen, then the immune system will just give the rapid response and pathogen and quickly produce the antibodies. This is called secondary immune response. So it is clear from this graph ki this is the blood antibody concentration which is being produced after entering of the pathogen in the body, that are also called as antigen. And this is the time period in days. So 0, 30 days, 60 days, 90 days, and this is the blood antibody concentration. So you can see ki this is the first antibody antigen exposure actually, where what is the meaning of antigen? Any foreign particle that has entered in the body, that is called as antigen. So body will give the primary response within, let's suppose this is 20 20. So the body will give the response within 20 days. 20 days. That is the this is the peak, and primary response is there. And the second exposure will be there. This is the primary exposure. This is the second exposure. So here the response is slow. It shows the narrow peak shows the response is slow, and this shows that whenever the second antigen exposure is there, the response produced is called secondary response, and it is more rapid and intense, and it is not more rapid. And now these two peaks are there which show the primary and secondary immune response. Primary response is slow and not very intense, and this is more rapid and intense. So primary and secondary immune response generally carried out with the help of two special lymphocytes, two special type of lymphocytes that are present in our blood, B lymphocytes and T lymphocytes. So B lymphocyte is generally are those lymphocytes which produce antibodies, or we can say the army of proteins. What is antibody? Antibody is actually the protein that these antibodies protect your body by these pathogens. So in response to pathogens, and our body is produce antibodies which is produced by B lymphocytes. Now what is the function of T lymphocyte? T lymphocyte themselves do not secrete any antibody, but they help these B lymphocyte to secrete the antibody. I am repeating again, the function of T lymphocyte. So T lymphocytes help the B lymphocyte to produce the antibodies. Now these antibodies are found in the blood. The antibody which is produced by the B lymphocytes are present in the blood. The response is also called, as they are present in the blood, so this is also called humoral immune response. T lymphocytes is found in the cell, so it is called as cell mediated immune response. Maturation of B lymphocytes takes place in bone marrow, and maturation is of T lymphocytes is takes place in thymus gland. So these two types of lymphocytes are there which help in the cell mediated or the first immune response is cell mediated or cellular immunity, which is by T lymphocytes. So T lymphocytes are being mature in the thymus and help the B lymphocyte. So four type of T lymphocytes are there: helper T cells, Th, killer T cells or cytotoxic T cells, and TK or TC, suppressor T cell or regulatory T cell, that is TS or TR, and memory T cells. Now this is the most important part of the chapter, antibody mediated immunity or humoral immune response, which is the antibodies produced by the B lymphocytes are present in the blood, right? So this response is based on the B lymphocytes, and these B lymphocytes secrete the antibody. Now we will go through with the structure of antibodies. These antibodies which are produced by B lymphocytes are present in the blood, and the response is called humoral immune response. Now antibody or immunoglobulin are the complex glycoprotein molecules which are made up of four polypeptide chains, two small called light chain and two longer called heavy chain. So here in the diagram you can see these two are the light chain, these are the light chain, and these are the heavy chain. This is the light chain, and this is the heavy chain, and this is the light chain. So in this way these chains are joined together by the disulfide bond. You can see there is disulfide bond present in between them. Disulfide bond here, these are present, disulfide bond, and these are the antigen binding sites. Whenever the antigen or foreign particle will bind here, then the antigen antibody reaction will be there, right? So this is amino terminal, this is carboxy terminal. So this is the structure of immunoglobulin. Two light chain and two heavy chain. So this is also called as H2L2. This is also called H2L2. These two chains held together by which bond? Disulfide bond. Now different type of antibodies are produced. Five type of antibodies are generally there that you can see: IgG, IgA, M, E, D. So what is the function of G? Is to protect the body fluids. A, protect the body surface. M, protect the bloodstream. E, mediate the regional hypersensitivity, and D, basically B lymphocytes, control the B lymphocyte. Function of antibody: just do these three functions like agglutination, opsonization, and neutralization. What is agglutination? Whenever the antibody, this is the antibody, Y shape structure is there. You can see this is the Y shape structure is there. Whenever this antibody is bind with the antigens at these sites, then antigen antibody reaction will be there. Antigen antibody reaction will be there, and this process is called agglutination. So basically this process will destroy the pathogen by the cell lysis. It will lyse the or break the cell. Now opsonization, so you just don't need to do in the CBSE, but still you can do if you want. Coating of bacteria with opsin or antibody. Whatever the pathogen is there, that is coated with, or antigen is there, that is coated with antibody. G and M, that facilitate the phagocytic cells. Means they will what they will do, they will just cover the antigen and phagocytic activity will be there, right? So this is called opsonization. Neutralization, neutralize the toxin of bacteria by attaching with them, so that bacteria will not be able to affect. Tissue grafting. So very often whenever the human organs like heart, eye, liver, kidney fail to function properly or satisfactory, there is one procedure called as transplantation. Is the only rapidly to replace that one or enable the patient to live a normal life. So graft, so the cells from the graft, any source of animal, the grafting can be taken from any source of animal, another primate or any other human being, that cannot be made, uh, otherwise it will be rejected. The graft, we cannot say कि blindly we can take the graft from anyone, or transplant we can take from anyone. It should be from compatible type of donor, right? Tissue matching should be there, blood group should match, and before taking any graft we should take care of all these things, कि tissue is being matched or not, blood group is matched or not, right? So in otherwise it will be rejected by the body, because body has the ability to recognize the self and non-self, right? This is the natural ability of the body, so the graft rejection will can be there, right? So the graft reject, to avoid the graft rejection, we should take care of these two things: tissue matching and blood group. Now, active and passive immunity. What is active and passive immunity? This is very important to know. Active immunity is actively produced by the immune system of host, and passive immunity generally passively received by the host, and host immune system does not participate in it. It is taken from outside. Inactive immunity, this the induced by infection. This activities actually, this immunity is induced by the infection with the immunogen or vaccine, and this is induced by introduction of ready-made antibodies, directly antibodies. Vaccines are not introduced, direct ready-made antibodies are produced. Immune response durable and effective in case of active immunity, and immune response is short-lived and less effective. And basically immunity develop only after a lag period. Lag period is that period in which the microorganisms grow, and it takes several weeks to develop, and its action is fast. And first of all the microorganism will grow, and then the vaccine. What is vaccine? Actually the vaccine is containing the pathogen in weak or inactive form. So it takes time to develop. Action is fast and immediate. Immunological memory is present. Subsequent challenge with booster dose is more effective, and no immune memory will not develop in case of passive immunity. Subsequent administration of again antibody will be there. अगर किसी को इन्फेक्शन होगा तो उसे पैसिव इम्यूनिटी के लिए वापस से वो एंटीबॉडी, रेडीमेड एंटीबॉडी लेनी होगी, जबकि यहाँ पर एक रिस्पॉन्स और मेमोरी डेवलप हो जाती है. एक्टिव इम्यूनिटी लॉन्ग लास्टिंग है, जबकि पैसिव इम्यूनिटी लास्ट फॉर फ्यू वीक्स और मंथ्स. यूज्ड फॉर द used for prophylaxis to increase body resistance, and जैसे कि फॉर एग्जांपल वैक्सीनेशन यूज फॉर ट्रीटमेंट ऑफ acute infection like colostrum, like हाँ yes, this is the very good example of passive immunity, in which the newly born infant is given the mother's milk, which is called as colostrum, which is thin yellow milk, very nutritious in nature. So colostrum is provided to the infant in that case, right? So what is the principle and mechanism of vaccination? Here preparation of antigen is there in the form of vaccine, in which the inactivated or weakened pathogen is taken, that is introduced in the body, and vaccine generate the primary first encounter of the vaccine by to the body generate the primary immune response and memory, B and T cell. So B cell will produce antibody, T cell will create memory. Now re-attack by the same pathogen or subsequent pathogen will lead the B lymphocyte to produce antibody by the help of T lymphocyte, which already has the memory for that one, right? So this is all about the active and passive immunity.